Excellent. Thanks, Renee, for the intro. As you can tell, I'm not Bo Bonnell. Bo was unable to be here with us today. If you don't know me, my name is Andrew Hansi, and I'm a research fish biologist with the USGS Great Lakes Science Center. And I have the pleasure of introducing our speaker today, Taylor Brown. Taylor is currently a PhD candidate at Cornell University, where she's studying cisco and lake whitefish recruitment across all of the Great Lakes, which is what she's going to be presenting on today. Her presentation very nearly represents the culmination of her dissertation work, as she will be defending her PhD in just about a month. Prior to her PhD, Taylor earned an Associate of Science degree from Northwest State Community College in Archbold, Ohio, followed by a Bachelor's of Science in Evolution and Ecology from The Ohio State University, where she studied predator-prey behavioral ecology with Stu Ludson and the late Mike Fraker. Taylor then went on to Cornell University, where she earned a master's degree in natural resources working with Lars Rudstam, Suresh Sethi, and Brian Weidel on larval cisco and lake whitefish distributions in Lake Ontario. Taylor, thank you very much for presenting for us today. And with that, I will hand it off to you.
Awesome. Thank you. Hi, everyone. Thank you all for being here. This is really exciting. So this, I realize, is a presentation that is 3 years in the making because I will be presenting 4 out of 5 of the chapters of my PhD. So we're going to try and do that in about 45, 50 minutes and hopefully learn a lot of things along the way. But yeah, like Andrew said, I'll be talking about lake whitefish and cisco recruitment dynamics and drivers across each of the Great Lakes and Lake Simcoe as well. And I will start off with a lot of gratitude for all of the people that are behind this and behind me doing this research. So I have an excellent team of co-authors and collaborators that make up this cross-basin project. And this research was really only possible due to many data providers that have been collecting decades of long-term data to that power every part of this analysis. And I want to acknowledge that funding for this came from the Great Lakes Fishery Commission. So these 2 fish are the stars of our show today. We have cisco up top, which is our pelagic planktivore, and lake whitefish at the bottom, our demersal benthivore. These are, of course, both coregonines. I'm going to skip most of the background on this, given that these are coregonines in the coregonine science webinar. And one of the biggest concerns we have and knowledge gaps for these 2 species is understanding why populations of both species are experiencing sporadic, poor, or declining recruitment across the Great Lakes, with a few notable exceptions. And so really needing to understand the drivers of the recruitment variability we're seeing, both when recruitment is declining and for those few exceptions where populations are increasing. And there really isn't a clear through line that we can point to for what's driving these patterns among lakes for a given species, or even within some lakes where we see population trajectories for these 2 fishes diverging even when they're occupying the same spawning habitats. And so the big argument of this is that by conducting cross-lake and cross-species comparisons, we can disentangle some of those drivers of recruitment variability, both within and across systems. So what do I mean by that? So first off, this is just a smattering of some of the factors that have been implicated in lake whitefish or cisco recruitment. And the Laurentian Great Lakes share a lot of fundamental similarities, both in their geology and their climate, but there's also really important differences among lakes along various ecosystem gradients and even physical gradients that matter for the fish that live in those ecosystems. And not only are there gradients across lakes, but also the lakes have experienced uneven ecosystem change through time. So in this way, we might expect that different lakes would have differing recruitment dynamics and trajectories, and we can use that to our advantage. And the other side of this is the cross species. So lake whitefish and cisco share generally quite similar early life histories during their embryo and larval stages, but those diverge when they become juveniles, and lake whitefish transition to benthic habitats and cisco pelagic. And because recruitment is generally thought to be regulated during early life stages, by comparing recruitment dynamics across these two species that have shared versus divergent parts of their life history, we can get better insights into when and where recruitment bottlenecks are occurring. But in order to make these kinds of comparisons at this scale, we really need comparable information and methods across systems. In other words, the ability to make apples to apples, very direct comparisons. And so I'll be touching on these two big research aims during this talk today, looking at recruitment dynamics up top and then drivers down at the bottom. And so, indexing recruitment by generating year class strength indices that are comparable across systems and between species, and characterizing recruitment dynamics across space and time. And then moving to the mechanisms or drivers of that by identifying the suite of biological and physical drivers of recruitment and quantifying their relative importance across ecosystem gradients and through time. And so we'll start off with the first one. So we searched high and low for any and all time series that could reliably inform the year class strength of cisco or whitefish through time, looking for long-term time series of catch and age that had reliable detections and consistent methods through time. We ended up with a final data set with 38 long-term surveys of catch and age data, a mix of fishery independent and fishery dependent surveys, and using a diversity of sampling gears, but mostly gill nets and trap nets. We are really, really happy with the temporal coverage we got from this data set. So our combined time series spans a sampling period of over 50 years, with a mean survey duration of 24 years, and with over 1 million unique records of age and other biological data for lake whitefish and cisco. And I will note here, we did end up finding much more data for lake whitefish than cisco. And just to give you a sense of the spatial coverage of this data across the Great Lakes and Simcoe, this is just on the left whitefish and on the right cisco, and where we have geo-referenced data from where these samples were collected. We do have additional data available at larger jurisdictional scales, like at whitefish management unit levels. But just to give you a sense, and to point out that cisco are extirpated from Lake Erie, so we didn't have any data there, and I won't be talking about them anymore in Lake Erie. As for how we actually chose to index recruitment, as I mentioned earlier, we were using year class strength as our index of recruitment. So this is the relative abundance of a year class or cohort based on repeated measures across multiple ages and sampling years. So this is fundamentally different than something like a single age recruitment index or like a stock assessment output. Getting a little more specific, these were longitudinal mixed effect regressions of relative abundance for each cohort through time, specifically when they first fully selected the gear. And so this is a really flexible method that allowed us to adjust the age ranges over which we were indexing year class strength based on the actual survey and when it was reliably starting to catch fish. And so this allowed us to account for the effects of different surveys, gear size selectivity, and changes through time, including even sampling effort. And this was a really great way for us to integrate really the best available data across all of our study lakes through time, including both fishery independent and dependent data in the same model. Importantly for our purposes, it allowed us to make standardized comparisons across our different lakes and between species where we have, for example, really different population sizes among these lakes. And I'll just take an opportunity to note that one of the things that was really apparent during this process is that the vast majority of long-term survey data across these lakes do not reliably observe either of these species before about age 3. And so we're going to be looking and talking about recruitment to the adult population for all of this. Before I start throwing data at you, I'm just going to walk through what the figure looks like for year class strength. So we'll have cohort on the x-axis. Okay, my laser pointer here. cohort on the x-axis, and relative year class strength on the y-axis. So these are all normalized, standardized. So the 0 line, it's not actually 0, it's the long-term mean relative to each model. Anything above that is going to be above average year class strength, and anything below that is below average year class strength. So we're really comparing the variability in year class through time among these lakes. So zooming out, we estimated year class strength using 11 separate models, so 2 species times 6 lakes, except for Cisco and Lake Erie. And these are lake-wide models, so using all data available within each lake. We were able to estimate year class strength for cohorts spanning 1956 to 2015. So here's what those data look like. I am going to walk through some of the key findings that we had in terms of the year class strength dynamics. I haven't put the citation up yet, but this is published open access. So you can find this and look through all of the details on your own time here. So first off, we found that year class strength was not synchronous or correlated between each species in any lake. So a high year class for Lake Whitefish in Lake Huron did not correspond to a high year class of Cisco in the same lake, for example. And that was consistent across all the lakes that we had data for both species on. In terms of looking across lakes for a given species, so starting with Lake Whitefish here, we found that on average, Lake Whitefish year class strength was positively synchronous among lakes. So a high year class in Lake Michigan corresponded on average to a high year class in Lake Ontario. So these are based on pairwise correlations. And so this wasn't true for every lake. You'll note that I have this big X here because Lake Superior was our odd one out in that it was not significantly correlated with any other lake. But in general, we did see this synchrony for Lake Whitefish. We also wanted to look at trends through time. And so we saw that Lake Whitefish year class strength in all 6 lakes was above average during this about 15-year period from 1980 to 1995. Those years aren't exact. But that after that period, there was no longer this shared temporal trend among lakes. The lake-specific trajectories diverged. Moving on to Cisco, we found that Cisco year class strength was not synchronous among lakes, either positively or negatively. They were just unrelated. And you'll also see that compared to especially the Lake Whitefish year class strength estimates, there's a lot more variability, up, down, up, down, up, down. And while that's most pronounced in Lake Superior, we do see that in all the other lakes. And so looking at that temporal trend again from that analysis that I showed for Whitefish, but this time for Cisco, we did not really find a lot of important trends through time, except for in a couple of lakes where we've seen pretty sizable differences. So Lake Michigan with a recovering population and Lake Simcoe as well, a recovering population. But for lakes like Superior and Ontario, those high boom and bust years sort of cancel each other out through time, at least within the temporal scope of our analysis. So we were really, really happy with the data coverage that we got, like I said, in terms of being able to reconstruct long-term year class strength for these species. And I think this is a really powerful information product for understanding the year class strength dynamics and drivers for these 2 species, well beyond what I'm going to talk about for the rest of today. But we also had a lot of new questions that emerged after doing this. So, you know, as I've said before, I'm not going to restate all of these, but we really had a lot of questions about what's driving some of these patterns we are seeing, not only within a given species, but between the 2 species, given that we saw really different recruitment dynamics, both in terms of the synchrony and in terms of things like periodicity and patterns in those dynamics. So what I'll be talking about for the rest of the day is moving on to trying to answer those questions. So we spent a while working on synthesizing hypotheses to actually interrogate with data, with really this emphasis again on comparisons between species and among lakes and taking advantage of those comparisons. So we did a lot in terms of empirical evidence in the white and gray literature, ecological first principles, and professional opinion, and including holding a virtual workshop with 57 participants. This paper is also available open access. And I'm not going to walk through every single one of these because, as you can imagine, there's a lot of hypotheses floating around, not only within each lake, but within each lake for each species. But I'll hit some of the high points. So the first one is sort of the no duh hypothesis, but just to set the stage, that we have a complex suite of biological and physical processes that interact to regulate recruitment of each species. So no surprises there. But processes regulating cohort size and survival during the first year of life are most important for determining year class strength or recruitment to the adult population in this case. So this ties back into the idea of when is recruitment set. And the idea is that recruitment is thought to be set by age 1 for both of these species. And so we can hone in on that first year of life to really understand what's driving this recruitment variability. And importantly, that the relative importance of driver-response relationships, or more specifically, the relationship between these biophysical drivers and year class strength in terms of their directionality, positive, negative, or something more complicated, and the magnitude of that effect varies among lakes that span ecosystem gradients through time in response to climatic and ecosystem change, and between species based on their differing life histories. And so I'll walk through a few of these in terms of getting a little more specific about putative biophysical drivers and comparing and contrasting between species and among lakes. So this is coming from that virtual workshop specifically in terms of professional opinion, but a lot of the shared drivers between the two species, things that are hypothesized to similarly be important for both species, included these large-scale climatic processes like temperature and growth potential and ice cover and winter severity during egg incubation, affecting early life stages, in addition to things like growth and predation risk during the larval stage. Can larvae find enough food? Can they avoid being eaten? Those were thought to be similar for the two species. In terms of what might be different, we see that really for lake whitefish, we see the uniqueness in terms of their direct interactions with benthic ecosystems coming into play here, and especially with benthic perturbations that have affected benthic prey availability and quality, leading to declining growth and fecundity as being uniquely important for lake whitefish compared to cisco that don't directly interact with those benthos in the same way. For cisco, things like changing pelagic primary productivity was thought to benefit cisco recruitment in terms of having benefited from oligotrophication. A lot of discussion about competition with planktivores, which, pretty prominently things like alewife, rainbow smelt, non-native planktivores. But ultimately, that metapopulation biomass and diversity was a really strong limiting factor. Moving into the among lakes. So perhaps unsurprisingly, there's a lot of these shared regional climatic factors that come out, like ice cover dynamics and water temperatures during early life, but also things like spawning habitat quality, and again, interactions with non-native planktivorous fishes like alewife and smelt. But we also saw important themes emerge among lakes. So for the upper Great Lakes, a lot, and especially for Michigan and Huron, I'll say, the thought is that limited pelagic primary productivity has induced a recruitment bottleneck with low zooplankton prey availability for larval and early juveniles. But there's a really different picture than in the lower Great Lakes. In Erie and Ontario, which are more productive, it's not thought that zooplankton are limiting, right? So we see this stark difference, and things like spawning habitat degradation and metapopulation diversity were thought to be more important. Okay, so those were, a bunch of hypotheses, but let's actually test the hypotheses where we have the data to do so. So the goals here were to, identify which of our hypothesized biophysical processes best explain year class strength for lake whitefish and cisco in each lake, with the idea of comparing and contrasting those relationships, not only which ones are important, but also the driver response relationships, to be able to then infer mechanisms by which they actually act to regulate your class strength, and understand to what degree do conditions during that first year of life really explain recruitment to the adult population. And as always, really emphasizing that cross-lake, cross-species comparisons. So we went back and we looked for more data, this time focusing on biophysical processes hypothesized to be important during that first year of life. So, more precisely, egg deposition to the end of age 0, with a strong focus on long-term spatially extensive data that are comparable among lakes, or at least all of the Great Lakes. So our minimum data cutoff was 15 years, because when each year is a single data point, you need a lot of data. We ended up with a lot of variables. Obviously, you should not actually try and read anything in that correlation matrix, but we had a lot of data to work with, but we refined that global predictor set ultimately to use variables that best represented the actual underlying processes of interest. So we removed highly correlated predictors based on, again, ecological reasoning and practical considerations, like where we had better time series, fewer missing data, better spatial coverage, etc. So doing our due diligence to improve both statistical power and to enhance our ability to infer what's actually going on beyond associations in the data. I'm going to walk through at a high level the predictor variables that we ultimately included in this analysis. I think there's 12 or 13. I'll just note that unless otherwise specified, we use annual indices for all of these, because unless there is a seasonal index, all of them were correlated with the annual index. So for example, the first bullet point here, we have annual degree days. Both spring and summer degree days were highly correlated with the annual index. So while we could, in theory, model the spring index, we're really tracking interannual variability versus anything about spring in particular. So yeah, we have annual degree days, thinking about temperature and growth potential, fall wind speed, that was indexed during the period of egg deposition, and spring wind speed, so when larvae are emerging, thinking about physical disturbance. We have 2 metrics of ice cover. We have ice cover duration during egg incubation, so that's the cumulative number of days. And then we have ice cover variability, which is the number of times that ice formed and then broke up throughout a season. So trying to get at stability or lack thereof in ice cover throughout a season, because there's a lot of interest in ice as in terms of protecting from physical disturbance. I'm trying to get at that more than just a how severe was the winter metric. And water level, which determines spawning and nursery habitat inundation, and also may indirectly also mediate things like nutrient inputs during high water level years. Coming on to the other half of our predictor variables, we have adult relative weight. And so this is a metric of physiological condition and energetic status that integrates multiple potentially important processes, things like growth and density dependence, and is really responsive to ecosystem shifts, especially changes in prey availability. That's one that has multiple potential underlying mechanisms, but has shown to be important for especially lake whitefish. We have Secchi depth, which we have as our indicator of biological productivity. I'll note that this was highly correlated with things like total phosphorus, chlorophyll, but had the best time series. So we have that in here. We have things like our planktivore index, which is a combined index with alewife and rainbow smelt, our non-native planktivores. We have round goby biomass. We have, and then we have metrics of prey availability. So total zooplankton density and total benthos density. So just to give you a sense of what these data look like, I won't spend too much time on this slide. This is just our predictor variable time series by lake. What I really want to point out here is that you might be able to see that the time series length, so how many years of data back in the past we can go, varies a lot between different predictors. So we have, for example, degree days where thanks to gridded climate models, we can go pretty far back in time. But things like total benthos density were limited to about 1998 forward. So having our predictor variables, we started to parameterize generalized additive models, or GAMs, that were lake and species specific, because preliminary analysis made it really clear that there was differing relationships going on. So a little bit of a spoiler alert for some of the results. And importantly, we parameterized a suite of models comprising predictors that varied in time series length. So I'll spend a little bit talking about why that's important without getting too much into the weeds. But what I have on the screen here is just for each predictor, I have box plots of the observed distributions through different time periods. And all I want you to take away from this is that the observed distribution of not only our predictor variables, but our year class strength estimates has really changed through time as the ecosystems have changed. And so depending on over which time period we're modeling, we might see really different distributions of these values that's going to affect what kinds of inferences we can draw and what kinds of relationships we can detect. So, you know, for these sorts of modeling exercises, if we wanted to model these all into one big model, we're limited by our shortest time series, which is, you know, 1998 forward, which, you know, in a practical sense, isn't a lot of data, but also in a ecological sense, we're limited to the post-Dracenid period and in a period of only low year class strength for many of these lakes. So, you know, for everything that I just said, our solution to that was to parameterize these three different sets of models, because we wanted to be able to look at all of these variables to the fullest extent that we could, while, you know, optimizing our compromises, right? So we're going to focus on drawing inference from all of these simultaneously, but we have our longest time series models, our max time series models that are, you know, 30 to 40 years. On the other end of the spectrum, we have our maximized covariates model. That's all of the variables in one model, limited to about 15 to 20 years. And we're able to include a lot of those biotic predictors that have, unfortunately, just systemically shorter time series than our abiotic predictors. And then we have our intermediate model in the middle. So all you need to worry about for this is that I will be showing, you know, another layer of models, but you can focus on drawing inference from all of them simultaneously. So, you know, then we applied model selection techniques to actually identify the subset of those predictors that best explained year class strength, and retained the predictors that were top ranked and parsimonious to fit and interpret for our final models, and using those to evaluate those driver response relationships and make comparisons among lakes and species. Okay, so what I'm showing you here is the performance of those final fitted reduced models with only those predictors that were most important. I have lake whitefish on the left and cisco on the right. On the x-axis are each lake. On the y-axis is the model R squared, or the percent variation explained. And I have, you know, different columns for each of those three different models I was talking about. I have an arbitrary dashed line at 50%, just so you can get an idea of where we have, models that explain a lot of variability versus not a lot. And this is also my cue to say that we did not model year class strength for Cisco in Lake Michigan or that final model for Lake Ontario because we dipped below our minimum sample size and we saw that that had really important impacts on the models. They just weren't robust enough to reliably model. So I'm going to focus this in on Lake Whitefish. One of the first things that you might see is that we had really great explanatory power for a few lakes like Huron, Michigan, and Ontario. And we had some lakes like Lake Superior where we had consistently, much worse explanatory power, and Erie and Simcoe somewhere in between. I'm going to start showing you data, but I'm going to build it there since there's a lot going on here. We're going to start first with Lake Whitefish, and then I'll move on to Cisco. So as before, we have year class strength on the y-axis. Each column is a different predictor variable. Each row is a different lake. These are arranged roughly in order of productivity because I think that helps with some of the interpretation. So they're not in downstream order, sorry. And then I have the observed predictor values on the x-axis. So this is just the observed data. So no biophysical model predictions yet, but each point is colored by cohort. So the darker the point, the farther back in time we're going. You can see that we didn't have data for every single combination of lake and predictors, but we got pretty dang close. Okay, so now I'm going to add lines for important predictors. So these are the fitted relationships between these predictors and year class strength for each lake. And the different colors correspond to those three different models I was talking about. So as much as I would love to just sit here and talk through every single panel with you all, we do not have time for that. So I'm going to walk through some key results, and you'll have to wait for the paper to be published to pour through every result. First off, one thing that jumped out right away was that ice cover variability came out as important in every single lake where we modeled it. We didn't have the data to support this in Lake Simcoe, like in terms of actually creating that predictor index. So unclear what exactly that would look like in Lake Simcoe, but at least for the Great Lakes where we had that data, it does come out as important. And it comes out as more important than ice cover duration itself. Those two variables weren't correlated. They both could have been selected in the models, as you can see for Lake Huron. But in general, we see ice cover variability was really important. And then for the other variables, these are just abiotic variables here that I'm showing. So same figure as before, but just zoomed in a little bit. We saw differences in the estimated driver response relationships that appear to map on really well to both latitudinal gradients and differences in lake morphometry. So first, I'll just focus your attention on annual degree days here. So one thing that you can see is that the range of observed degree days differs a lot by lakes. So we have Lake Superior, which was consistently our coldest lake, and that had a consistently positive association with increasing temperature. But as we get more southern and into smaller lakes, that relationship goes from dome-shaped to more strongly negative. And in some cases, we even see that this relationship gets more strongly negative through time as we go into our near-term models where there's fewer and fewer colder days showing up. The other things that we see in terms of wind speed and water levels is that we see differing directionalities among some of these lakes that we think, again, are related to differences in lake morphometry. One of the really clear important variables that came out was adult relative weight. So that came out as important in every single lake. And I will say in Huron, Michigan, and Ontario, the lakes where we had really strong explanatory power, this is where that is coming from. But interestingly, we see that we have pretty different relationships among different groups of lakes, right? So we have this almost positive with an asymptote relationship in those three aforementioned lakes. But then we have negative relationships with Superior and Erie and a dome shaped in Simcoe. And we can also see differences in sort of these longer term models, like our max time series models, and into our near term models, like that max Cope model, where the range of distribution of those observed values really shortens. And so, as I mentioned earlier, adult relative weight is a little tricky because it does encompass multiple potential mechanisms. And so we think that the negative relationships that we're seeing in Superior and Erie, especially Superior, might be due to things like density dependence and interspecific competition, or maybe for Erie, that one has had pretty large shifts in population structure through time. So that might be what's going on there. But in terms of Huron, Michigan, and Ontario, one clear thing that we saw based on some historical Lake Ontario data is that adult relative weight really maps on very strongly to long-term benthic prey, total benthos density in Lake Ontario. So this kind of historical benthos data isn't available in every lake, and so we didn't include this in our primary models. But we see on the left here, this is that historical long-term total benthos density from this data paper by Luba Berlikova and Sasha Karatayev, and our lake whitefish adult relative weight values in Lake Ontario. And they model quite well together. And I will say that in terms of how these models are actually behaving, at least for Lake Ontario, adult relative weight switches out with this long-term total benthos density. And we know that similar effects have happened in the other lakes where adult relative weight had this really same relationship in Huron and Michigan. But, here I have box plots with observed distributions of data. On the right is that near-term total benthos density from the standardized survey from all of the Great Lakes. And in this Max Cove, our final model, we're sort of left with this post-Dracaena view of what these benthic ecosystems look like. But with these long-term data, we have the ability to go farther back in time, and we see that mapped onto our adult relative weight. Last big theme for lake whitefish, before I have to move on to Cisco. In general, we found that lake whitefish year class strength increased with variables that were related to increasing productivity. So things like increasing secondary depth, increasing zooplankton density, and even things like increasing planktivore biomass, which in this we really think is a productivity signal. And then we didn't see a lot going on in terms of this total benthos variable. We did see a positive relationship with Lake Huron, but ultimately this is across these lakes, we think we have limited ability to detect major shifts in total benthos given the very near-term limited scope of these data. Okay, moving on to Cisco. Yeah, in general, we got pretty good model performance with these, with some exceptions, like some of the longer term models for Lake Simcoe. Again, I'll start by showing you the actual observed data. We have a little bit less to work with Cisco. I'm showing the Lake Michigan data here, but again, we didn't model it. So again, adding on those important variables, and I'm going to walk through some of the key trends since we don't have a ton of time. The first thing that popped out is that we saw more differences than similarities among lakes, especially given some of the strong similarities we saw among lakes for the same analysis for lake whitefish. And ultimately this makes sense given that we saw that recruitment dynamics were not synchronous among lakes. There's different drivers going on. And one of the themes that does emerge, though, is that cisco year class strength often decreased with increasing productivity and variables that were related to that, which is the exact opposite of lake whitefish. We have one exception, which is Secchi depth in Lake Ontario. But otherwise, we see things like a negative relationship with total zooplankton density, which we think is tracking on to decreasing zooplankton densities as oligotrophication proceeded. And things like a really strong threshold relationship with Secchi depth in Lake Simcoe. We also saw things like a negative relationship with adult relative weight in Lake Superior that was quite strong. And then I just wanted to take an opportunity to directly compare lake whitefish and cisco. So lake whitefish is in orange, cisco is in purple. And I'm showing side by side the relationships from that max time series, that longest term model. And what I hope is apparent is that in general, there were very few instances where the same driver was retained as important within a given lake for both species. And most of the time when that did happen, they were going in completely opposite directions. That's not universal, but that became really apparent early on. Okay, so that's the end of my results. Now it's time to take a second to sort of walk through what this all might mean. So again, I'll start with lake whitefish here. One of the biggest themes that we see throughout the results that we have here on both the dynamics and the drivers is that those recent cross-lake trajectories that have diverged among lakes map on very well to ecosystem shifts related to dracynid mussels, things like especially benthic food web perturbations and declining pelagic productivity as well. And we see differences based on the extent to which these shifts occurred across trophic gradients. So we see that in deep oligotrophic lakes like Huron, Michigan, and Ontario, where quagga mussels were coincided with things like collapse of diaporia, a key prey item. Benthic prey availability appears to be limiting recruitment. But this is a pretty stark contrast to our productive and relatively shallow lakes, Erie and Simcoe, that have zebra and quagga mussels, but have not had the same level of impact, at least on lake whitefish, in terms of collapse of their benthic communities available to lake whitefish. And Superior, of course, remains stable, at least for now, not having dracynid mussels established. We saw with the importance of degree days and ice cover variability that we already are seeing signals of climate change in these data for lake whitefish, with things like that intensifying negative relationship with temperature that we saw in some lakes. And this being an important broad scale regional driver of year class strength in the system, but that has differing consequences across these different lakes. The impacts of increasing temperature are very different in Lake Superior versus Lake Erie, for example. And then trying to get at this question of what was driving that shared synchrony in the 80s and 90s for lake whitefish. With all of these data combined, it seems that across these 6 lakes, ecosystem conditions seem to have been in that sweet spot of improved water quality from really eutrophic conditions that still maintained intermediate, relatively high primary productivity prior to oligotrophication really accelerating, favorable climatic conditions prior to things like global climate oscillations, and for many of our lakes before dracynids became established. And so, to put it bluntly, these same conditions no longer exist today in some of our study lakes because of climatic and ecosystem shifts. And so I think that's really informative for how we move forward in the future and have future expectations for recruitment capacity in some of these lakes. Moving on to cisco, we saw that these boom and bust recruitment dynamics were consistent across all of our study lakes. and are likely intrinsic to at least Great Lakes cisco populations. In terms of what might be driving that, we're hypothesizing that that negative relationship we saw in Lake Superior with adult relative weight, which is our largest population of cisco, suggests that interspecific competition and density-dependent effects might be important for driving that. And that's previously been hypothesized for Lake Superior as well. In terms of some of the drivers of both the asynchrony that we saw and just in general, we don't have as clear of a smoking gun that we did for Lake Whitefish with some of those benthic perturbations. Perhaps because we have fewer lakes to look at or fewer years of data, but we have some ideas. It might simply be a lack of spawning stock biomass among lakes other than Superior to capitalize on some of those prime environmental conditions. But I will say the differing directionality that we're seeing in some of those abiotic factors doesn't support that hypothesis. Instead, thinking more about some context dependence across these ecosystems that span different physical and biological gradients, or maybe we have populations that are responding differentially to these regional climate effects. For those of you that have working familiarity with cisco across the Great Lakes, you might know that populations can have really different spawning habitat preferences and nursery habitat preferences for their larvae. And so maybe there's, that's a consequence of differences in deep versus shallow spawning populations, for example. We found that in general, abiotic factors were generally more important than biotic factors, but as I've said, not a lot of consistency, and that oligotrophication appears to have benefited cisco. So, our results that show that things are really different across lakes for cisco really affirms that restoration planning should be tailored to each lake, which thankfully is already a guiding principle of the coregonine restoration framework. But importantly, I think this really drives home that caution is warranted when applying inference, whether that's in their ecology or what interventions might be effective from one lake to another. Most of our understanding of cisco ecology in the Great Lakes comes from Lake Superior, which makes sense. It's where we have decently large remnant populations. But I think we might have to be careful about strictly inferring cisco ecology in the other lakes in the context of what cisco and Lake Superior look like. And then putting this together in terms of Lake Whitefish versus cisco, we really did see very different recruitment dynamics and drivers, which suggests pretty clearly that there's different recruitment bottlenecks. And there's a couple of clear themes that emerged in terms of different responses to oligotrophication and benthic perturbations that directly and disproportionately affect Lake Whitefish compared to cisco. And all of this collectively indicates that bottlenecks responsible for the divergent recruitment trajectories between the two species likely occur by the end of or after the larval stage when their life histories strictly diverge. And that's not to say other bottlenecks aren't important, but really thinking about specifically which ones explain the divergent recruitment trajectories we're seeing. And I will say, we are looking at recruitment to the adult population. And if we were able to look more closely at factors occurring during those shared life history periods, we might have seen more similarities. So, next steps, future research. This was an exercise, especially on the driver's side, of describing patterns in the data and generating hypotheses about the mechanisms that underlie those associations. But, there's still a lot of work to be done in terms of testing the hypothesized relationships we've inferred here. We have this cross-basin scale of analysis that was really important for detecting some of these overarching patterns, like looking at drivers across latitudinal gradients. But, there's still a lot of depth that we can obtain by looking at lake and within lake scales. And despite all of the data I've shown today, we were still limited in data. So as I've mentioned before, in terms of early life stage abundances where we can get at different questions, data at finer spatiotemporal scales like nearshore zooplankton would have been really interesting to look at and really underscores the need for continued monitoring and historical data rescue, because some of these do exist, they're just not available. So I'll just leave this up on the screen here because I know I'm already over on time where I wanted to be, but some conclusions and parting thoughts, and that I've already touched on a lot today. And yeah, it's an understatement to say that I've covered a lot of ground, but left a lot of the details unsaid. So I'm happy to answer questions and continue the conversation. Thank you all.
Thank you very much, Taylor, for an excellent presentation. I I was very surprised when I looked at the clock as you were rolling up and realized how much time had passed. It was a very engaging presentation, and it felt a lot faster than how long it actually was. So excellent job. And yeah, we are happy to field any questions from anyone in the audience. If you'd like to ask a question, please feel free to raise your hand or turn your camera on. You can also put questions in the chat, and we will try to keep up with those. Any questions out there? Oh, Dave, go ahead.
Great job, Taylor. Really enjoyed it. Very interesting. A lot to get your mind around when you're looking at all these different lakes and all these different factors. But, the whole time as you're going through this, I'm thinking about what are the implications for these reintroduction efforts like we see going on in Saginaw Bay and debated for Lake Erie and different places. So, what's your sense about that? Saginaw Bay is productive. I think ice cover has become more variable there. Some years it's really good, others not almost non-existent. Any insights that way?
Yeah, I mean, that's also a big question. I think, the first one that comes to mind, and I know that you've thought a lot about, is in terms of the Cisco boom and bust dynamic, where even under, good conditions, that, they're not going to be having consistent recruitment year after year. That just seems very apparent, at least on what we're seeing in these data under relatively contemporary conditions. And so, I think that that's really important in terms of calibrating your expectations for how long some of this might take. And yeah, I mean, I think just in general, I think, you know, beyond maybe like specific thoughts about Saginaw and Lake Erie in particular, I think just an appreciation is needed for what kind of environments these Cisco and are being reintroduced to and how they might interact with that environment, right? Like how Cisco may interact with the food web in Lake Erie is just fundamentally going to look very different and superior. And so without having any specific answer for that, I think thinking mechanistically and like with even when we don't have empirical data for what that looks like, putting on our like first principles hat and thinking about mechanistic relationships, I think that can be informative.
Excellent. Thank you, Taylor. I see we have a hand up, but first, before we go to Lisa, I'm actually going to jump into the chat here. I see Ed Rutherford put a question in there. Ed asks, with the introduction of round goby and its importance as prey to adult lake whitefish, have you seen a change in response of lake whitefish adult relative weight and a potential effect on year class strength?
Yeah, that's a really good question. I would have to look within just the time period of when round goby come in, but I would say that in general, it's not like we see in Huron, Michigan, and Ontario where we have these like very clear, before and after, high, low values. I don't think I've seen a lot going on in that low value in terms of recent increases, but I think that could be interesting because I've thought about the same thing. Yeah, that would be really interesting to look at closer. Thank you, Ed.
Okay, we'll go to Lisa.
Thank you. Great talk, Taylor. Hi. So, where to begin? There is a consortium of fisheries managers from Maine, New Hampshire, Vermont, New York that are interested in looking at coregonid dynamics and restoration and management in inland lakes. So, New York straddles the Great Lakes, and we have some work that's gone on in the Finger Lakes as sort of a mesocosm. But state of Maine, they have a couple of really big lakes. But we're interested in putting together sort of a similar type of data. Like, what do we do looking at corrigona dynamics and restoration, particularly across these inland lake systems? And I mean, the Great Lakes are their own animal, but do you feel like this kind of an exercise would be applicable to inland lakes where you have huge size variation in, like little tiny Adirondack kettle lakes that house, brown whitefish versus, I don't know, some of the big, like Lake Sebago in Maine that are huge, complex dynamic systems? Do you think this type of an approach or even the parameters that you've identified would be something we could start to dig our teeth into for these Northeast lakes, these inland lakes?
Yeah, I mean, my answer is absolutely. I think that that would be an excellent approach. As you have these lakes that are spanning, this landscape of lakes where you have variation in climate across that region, but also one of the reasons why we included Lake Simcoe in this analysis, which while still being a really large lake is a very different size than the Great Lakes, is because it's thought that the relative importance of density dependent versus density independent processes vary a lot along that continuum. And so you might, by comparing across those lakes that are really different sizes, like you mentioned, that might become more evident in terms of, like, I know I've read literature out of rainbow smelt predation of lake whitefish in some of those main lakes. And that might be really different in a large lake versus a small lake. You're more likely to run into data limitations, but depending on what kind of data you have, there might be paths forward there. But that would be that's my only hesitation of, how much data can you actually look at some of these long-term patterns?
Right. I think that the data limitation, as you said, is going to be the problem, but maybe moving forward, we could all start to pick some of your very important parameters that you've already identified and said, okay, everybody start measuring this. So, so I will be tracking you down later for more stuff. Thank you.
Thank you, Lisa. We have another question in the chat from Charlie Roswell. Hi, Charlie. Sorry if I missed this, but did you incorporate spawner biomass or abundance, Taylor?
Yeah, thanks for the question. I did not mention this. No, we did not, because we don't have estimates of this across all of our study lakes or between both of our species. We looked into various ways of incorporating this using the survey data that we have, but it was just painfully obvious that we were seeing, like, most of the variation in those estimates were an artifact of the surveys themselves and things like measurement and like observation versus reflecting an underlying trend. So we were unable to do that. But I will say that the year class strength estimates themselves are, because they're standardized, it's really focusing more on the variability versus the absolute numbers. So yeah, definitely a factor that I would have loved to incorporate in terms of some of these factors might matter more or less in high versus low spawning populations. Absolutely. But that would probably have to be saved for a lake specific analysis where we have the data to look at that through time. And we just did not have that in most of our lakes, unfortunately.
Excellent. Thanks, Taylor. Another question from Elizabeth Bradshaw. Have lake whitefish begun to feed on dreissenids at all?
Yeah, from what I understand, they will happily eat them, but it might not have any real benefit in terms of nutritional quality for the amount of time it takes to digest them. And there's some outstanding questions about things like thiaminase that I know some folks are working on. I don't know what the answers to that are. So yeah, we did not include that in terms of our benthic prey availability predictor index, because it's just thought that they really aren't, even if they are available as prey for lake whitefish, They're not quality prey by any means.
Very good. Any other questions? Again, please feel free to use the raise hand function or turn your camera on, or continue to submit questions in the chat. Corey.
Hello from Ann Arbor. Can you all hear me?
We can.
Oh, good. I'm coming at you from the conference room down here, so I don't know how it's functioning, but great talk. I love this talk and hearing about this work. I had a quick question. It's so cool about the cross-lake comparisons with all these variables. Is there any way to fine-tune this within one lake, or did you do any investigating? Like I'm thinking about Lake Huron, where there's still extant cisco populations in certain regions of the lake, like the North Channel, versus like the main basin and southern Lake Huron. So within lake fine-tuning, any thoughts on that ability with this method?
Oh yeah, absolutely. Basically, that's the next step, to see to what degree we have data to support doing within lake analyses. So not just going down to a single lake, but looking within lake. That is pretty data hungry. And so we're gonna, I'm optimistic, but it'll be a little bit of a battle, but I'm hopeful that we can at least get to the point where maybe we have a couple of regions within each lake that we have robust enough data to have confidence in that. And then compare that to, if not to each other, then to the lake-wide estimates that we have here. Maybe things will track, maybe we'll see that things are very different. And our data coverage it definitely varies by lake. Like we'll definitely be able to do that for Lake Superior, definitely for Lake Whitefish in like Huron and Michigan, etc. The Cisco data are a lot more limited. So we will have to see, outside of Lake Superior, what we can do with that.
Amazing and super exciting. Thank you so much.
Yeah, thanks, Corey. Any other questions? Okay, well, not hearing any additional questions, I think we can go ahead and wrap up. Once again, thank you very much to Taylor for an excellent presentation, and thank you all for attending. And we will see you at the next webinar.